What if I told you that there was a model that you could use that could be useful for human health as well as increasing food production? That's exactly what we're trying to do by studying pregnancy using sheep as a model. They share a number of key features of pregnancy and placental development with humans, making them ideal for studying complications that arise in humans like preeclampsia and placental insufficiency and things like fetal growth restriction. At the same time, they're agriculturally important animals, and so what we can learn from them is highly adaptable to other livestock species, especially cattle, which share a number of similarities with sheep in terms of reproduction and how the placenta forms and functions. At the heart of our research is the placenta. I am truly fascinated by the placenta. I think it's an amazing and unique organ. It's the only example that I know of where an individual forms an organ as an adult. Most of our organs are formed when we're all in utero. The communication between embryo and the cells of the embryo and mom's uterus and the inner layer of her uterus, which have completely different genetic makeups. And so there's this microenvironment where we have to make sure that the immune system of mom's cells are not attacking and killing the embryo, and these cells work together to completely make a brand new organ that is the placenta. And that placenta is absolutely vital. So what's happening during placental formation and how it functions during gestation can have not only an impact on just in utero health, but lifelong health of both livestock and humans. Yet despite the importance of this amazing organ, the placenta, it's actually one of the least understood organs, and particularly if we look at how the placenta forms. And a lot of that stems from the fact that it's quite difficult and impractical to study how the embryo attaches and implants in the human uterus. And so there's always this need for an animal model to try to study and replicate what's happening in humans. Our research is focusing on a specific chemokine pathway. So chemokines are these small little peptides that are typically made by immune cells. The particular chemokine that we study, called CXCL12, it's made by immune cells, but it's also made by non-immune cells, and specifically it's made by cells of the embryo, which are called trophoblast cells. And what's interesting is these trophoblast cells are secreting this chemokine, CXCL12, and it's then going and talking to cells in the uterus that express its partner, which is called CXCR4. And so when CXCL12 and CXCR4 form and come together, that's how they cause some type of biological response. One of the innovative things about our research is that we're able to specifically study and manipulate this chemokine pathway during that time that the embryo is attaching to the uterus in the live animal. We've shown that by blocking CXCL12 during this early window of implantation, that we have negative consequences to placental development with drastic changes in vascularization and embryo attachment. What's interesting is these are mimicking the root causes that we see that leads to things like preeclampsia and placental insufficiency in women. So vital to the success of our research program is the students. It takes, as they say, it takes a village to raise a student. It takes a village of students to do these research projects. But I've been very fortunate and blessed to have just an amazing group of both undergraduate and graduate students that have really come together and worked well together as a team. And typically we just all share the load. At the same time, students are getting lots of just hands-on experience that they wouldn't get otherwise. Ultimately, this work does have a dual benefit. By studying this early signaling of when the placenta develops in an animal model, we can gain a greater understanding of just overall biology of how the placenta forms, and at the same time, we're able to develop therapeutics or methods to try to enhance reproduction and reduce pregnancy losses for both women and livestock and dairy producers.